Climbs rapidly. Well, it is 7 a.m. Our speaker this morning, Tomas Mustelin, I hope I'm saying that somewhat correctly.
Yes.
Professor and the chief of rheumatology. What strikes me for us as allergists is a very interesting topic, because since we're constantly focused on eosinophils as mediators of, and I would substitute pulmonary or respiratory disease. Instead, we don't generally think of it as a mediator of rheumatologic disease and therapeutic targets for treating those diseases. So we'll let you go ahead. Thank you very much for joining us this morning, Tomas.
Thank you very much. Do you see all my slide in presentation mode properly?
I see your slides, and I see you as the only person live.
Okay, I hope I'm not the only one alive, but
we're good.
So thanks for the invitation, and I'm very happy to talk to you about eosinophils more from a rheumatological perspective today. I also have a little second part to my talk about eosinophils as therapeutic targets that I think is interesting to think about, more as an experiment to really figure out what the role of eosinophils is in a range of different diseases. So I don't have any disclosures from the last few years. And just to put this talk in a little bit of a larger context, about half of all our circulating immune cells, white blood cells, are granulocytes. The normal range is indicated here. Some people have exactly 50% of their immune cells as granulocytes. Some people have a little bit more, a little bit less. So the normal range is fairly broad. There's actually a circadian rhythm also where granulocytes vary in numbers in any individual across the day of 24 hours. So if, as originally was done by Paul Ehrlich in the late 1800s, if you stain blood with a basic and an acidic stain, hematoxylin and eosin, the H&E stain, which is routinely used, you will find that most granulocytes stain very little, and they are all referred to as neutrophils, while a small subset stain strongly acidically and are therefore called eosinophils. They love the eosin color simply. And another even smaller subset stain very darkly and are called basophils. And so I'm going to focus today on the middle granulocytes, which I think you are all quite familiar from your work with. So I think this slide is important. If you remember what drives eosinophils, what brings them in, what they do, and what they will secrete, you kind of in a nutshell can figure out what neutrophils do in disease conditions. So like all granulocytes, eosinophils are produced at a constant, fairly high rate. They have a short half-life in circulation, but they have a propensity to exit the bloodstream and spend most of their life out in tissues with half-lives that seem to be several weeks, if not months. So granulocytes are driven, their formation is driven by a number of cytokines, particularly GM-CSF and IL-3 as all granulocytes. And in addition to that, very uniquely for this subset, IL-5, and I will talk more about that in a little bit. Eosinophils are attracted to sites of relevant type of inflammation by chemokines, particularly the eotoxins, as well as RANTES. These work through the chemokine receptors CCR3 and CCR5. CCR3 particularly is fairly restricted to a smaller set of immune cells. Like all granulocytes, eosinophils are very good at getting activated and producing reactive oxygen species, including some that contain bromine as well as oxygen radicals and ions. They also make plenty of leukotrienes and prostaglandins and a whole range of interleukins and cytokines, as well as growth factors, including VEGF, importantly, and nerve growth factor, fibroblast growth factor, and platelet-derived growth factor, plus a number of other enzymes. And then more uniquely to the eosinophils, and actually a reason that they stain so strongly red in the eosin stain, is that their secondary granules have a very different content compared to neutrophils. So they have proteins like the MBP, major basic protein, and eosinophil cationic protein, eosinophil peroxidase, and a eosinophil-derived neurotoxin. Those are instrumental, I believe, in many of the immune responses where eosinophils play an active role. Recently, it's been found that eosinophils have another unique behavior that they can undertake. I suppose many of you have heard about neutrophils undergoing netosis. And it turns out that eosinophils can do exactly the same thing, except we have to call it e-etosis rather than netosis. So this is a reaction where the cells decondense much of their chromosomal DNA and spit out through a hole in the membrane this loose DNA that then forms an extracellular network of DNA with associated, particularly basic proteins. And it's believed that in, for example, bacterial infections, that this extracellular DNA network works to trap bacteria as well as platelets and many proteins involved in antiviral, antibacterial defense. So in the case of eosinophils, the composition is probably slightly different, but the same idea of extracellular DNA, which is super inflammatory and can cause vascular problems as well. So let me just point out that IL-5 is critically important for eosinophil development. So eosinophils express fairly uniquely the IL-5 receptor. I will mention towards the end that actually basophils also have IL-5 receptors. Otherwise, that receptor is not expressed on other immune cells. And eosinophils uniquely depend entirely on IL-5 for their development, as well as their accelerated development in the right inflammatory conditions. So eosinophils, I think honestly, are relatively poorly understood still as effector cells of an immune response. But clearly, many of the cytokines that they produce will contribute to tissue changes, tissue responses to an active ongoing immune response. So for example, cytokines like IL-13 and IL-4 will have a multitude of effects that we often think about as Th2 T-cell mediated effects, but eosinophils can make those cytokines as well. They make growth factors that can affect fibroblasts, epithelial cells, particularly in the airway, something you are probably all familiar with, as well as the vasculature and fibroblast differentiation into myofibroblasts, which are important in scar formation, fibrosis, airway remodeling, and of course the same things in other tissues and organs. So a key, I think, the initial key to the role of eosinophils in human disease have a lot to do with the number of them in blood. So as I said a moment ago, the the range of eosinophils normally in healthy human blood is less than 350 cells per microliter. And if the number exceeds 500, we call the condition eosinophilia. And if they are really increased more than 3 or 4-fold, so over 1500 per microliter, or 1.5 times 10 to the 9th per liter, we've referred to it as hypereosinophilia. So for a long time, that was known, and it was associated with a number of conditions, but it wasn't really clear whether eosinophils were just reacting to something or whether they participated in the pathogenesis or tissue damage associated with these conditions. And that's what I'm going to come back to a little bit later on here. There is also clearly recognizable tissue eosinophilia. So where the number in blood might be normal or only slightly elevated, but there's a great accumulation of eosinophils in certain tissues, often discovered by serendipity or looked for in certain tissues, but can occur in lots of different organs and tissues. So eosinophilia, when found in a differential white blood cell count, could indicate many different things. And here I have a list of those things. So eosinophils for a long time have been known to participate in responses against parasites, worms, fungal infections, which is kind of a curious finding in a way, because these are much, much larger pathogens than the eosinophil itself. But eosinophils have a tendency to adhere to the surface of these parasites or worms, and then use their secondary granule contents to try to make life difficult for these much, much larger adversaries. What you are all, I'm sure, very familiar with is eosinophilia as an indication of certain forms of allergic reactions, which can be to many different things, including drugs, as well as, of course, asthma, particularly of the endotype referred to as eosinophilic asthma, which I believe is a pathogenically, molecularly somewhat distinct form of the disease, which I will come back to also in a moment. There are a number of other conditions. So if you have a very large increase of eosinophils, but it isn't associated with any of the infectious or allergic asthmatic conditions, it is sometimes referred to just as a hypereosinophilic syndrome, which could be either acute or chronic. And it's important in these cases to try to figure out if there's something more behind it. So one of those things could be a adrenal insufficiency or early Addison's disease. So here the mechanism is fairly straightforward. Corticosteroids will suppress the production of eosinophils and their numbers in circulation. So if you have lower than normal levels of corticosteroids, eosinophils simply just increase for that reason. A more sinister type of hypereosinophilia is one that has to do with stem cell abnormalities, so early granulocyte mutations, for example. These can be either benign or frank eosinophilic leukemia, and of course need to be dealt with by a hematologist, oncologist. There are some cases of idiopathic, so there's an increased number of eosinophils for no particular other reason. And in some of these cases, there are mutations. So these conditions do not rise to the level of a malignancy and maybe aren't clonal, but still sometimes have to do with mutations in transcription factors or other more eosinophilic related pathways of homeostasis. And then a number of rheumatological conditions, of which the eosinophilic form of vasculitis is important, and I will talk a lot more about that in just a brief moment. And I have a number of references at the bottom of the slide. So for those who are interested and want to dig in a little bit more into some of the things that I'm mentioning, I warmly recommend looking up these papers, mostly reviews like this one, for example, in Nature Reviews in Rheumatology. Just a few things about tissue eosinophilia. So as I mentioned, this is when eosinophils are increased either by histology or other ways of enumerating them, typically in tissue samples, occasionally in tissue fluids as well. And eosinophils in blood can be normal. So tissue eosinophilia is most commonly seen in the heart, in kidneys, and of course in the lung. So we often talk about these conditions as more distinct clinical entities. And the eosinophilic vasculitis is one I will talk more about. I should probably say that eosinophilic esophagitis is, relatively speaking, more prevalent condition. Eosinophilic fasciitis, not unheard of at all. And eosinophilic vasculitis is really the type of tissue eosinophilia that rheumatologists, I can't say frequently, it's still a rare disease, but among the vasculitides, it's not unheard of at all to see that disease. This is a Venn diagram. I'm a very visual person. I like to sort of put things in cartoons and graphs to sort of conceptualize things. So what I intended to message with this slide is that the pathogenesis of many diseases have a great deal of overlap, share molecular features with diseases, and particularly atopic dermatitis, asthma, and COPD have a great deal of commonalities, for example, in Th2 immunity. Nevertheless, each disease also represents a range of different pathogenic mechanisms that I think have a sometimes more distinct representation, sometimes more overlapping. And so eosinophils play a role clearly in a subset of asthmatics and a form of chronic obstructive pulmonary disease, COPD, that is characterized by increased numbers of eosinophils, so lung eosinophilia. I think the endotype of eosinophilic asthma is a fairly well recognized one. Eosinophilic COPD is a bit more nebulous and perhaps represents a transition of eosinophilic asthma into a more chronic phase and maybe more COPD-like condition later in life. So diseases like eosinophilic vasculitis and eosinophilic esophagitis, of course, are entirely within the realm where eosinophils play a role. I'm not going to call them causative. I'm using the word participate in the disease rather than cause it, often because it isn't entirely clear. So let's move over to rheumatology then, and first say that there are many diseases that rheumatologists work with on a frequent basis where eosinophils can be increased. So fairly common rheumatological conditions like rheumatoid arthritis, systemic lupus erythematosus, progressive sclerosis, Sjögren's syndrome, and so forth, sometimes have increased eosinophil numbers. It is probably not hugely important in those diseases and may be related more to a broader immune activation and mobilization of granulocytes. So it's really the disease where eosinophils are clearly important is eosinophilic granulomatosis with polyangiitis, which I will talk more about. There is also a fairly important role in IgG4 related disease, and I'm going to show a quick cartoon from a review that we published last year, because it kind of illustrates how eosinophils in many conditions are thought to be engaged in the broader immune response. So in Th2 immune responses, type 2 immune responses, where the Th2 CD4 positive helper cell plays an important role, for example, in response to allergens or helminths parasites. It's relatively straightforward what happens. So Th2 cells are the main producer of IL-5, and if IL-5 is produced at enhanced, elevated levels for some time, it will accelerate eosinophil production, and eosinophil numbers will increase. There are also other mediators for eosinophil recruitment, which are not included in the slide here. So eosinophil mobilization and production in many conditions is driven by reactions that sort of look like this cartoon. In eosinophilic granulomatosis with polyangiitis, or EGPA, it is probably a little bit different. So this syndrome was originally described in 1951 by Churg and Strauss in Germany. It is usually referred to nowadays as EGPA, just because syndrome names with the discoverer's last names are a lot less informative, it is felt, than actually a name that describes what the disease looks like. So EGPA is one of the three main forms of what is referred to as anti-neutrophil cytoplasmic antibody associated vasculitis. So ANCA associated vasculitis, or AAV. So anti-neutrophil cytoplasmic antibodies are a set of autoantibodies that in a resting granulocyte would recognize the specific granules, so intracellular antigens. And these are proteins, particularly proteinase 3, PR3, and myeloperoxidase, MPO, and sometimes lactoferrin, that during the priming and activation of granulocytes get externalized out on the surface. And the autoantibodies can then bind to them on the surface of the granulocyte, as well as to the Fc receptors on the same or neighboring granulocytes, and cause an activation of the granulocyte. So these are the three main forms of this vasculitis syndrome. And I'm going to focus exclusively on the one to the far right, which is the eosinophilic form. So as you can see from the numbers, it is a fairly rare disease. Only maybe one case per million years in incidence, and maybe 2 to 20 or so cases per million. This is geographically variable. In some regions on the planet, the incidence is a little bit higher, and some a little bit lower, which has to do, I believe, with genetic predisposition, a number of alleles of genes that have a geographically uneven distribution on the planet. Interesting also, and unlike most other rheumatological conditions, EGPA, as well as the other forms of ANCA vasculitis, have a 1-to-1 gender ratio, males to females 1-to-1. In most diseases with autoantibodies or other manifestations, there's a female predominant distribution. So as the name says, there are three components: the eosinophilic. So EGPA is characterized by eosinophilia, and particularly tissue eosinophilia, and granulomatosis, which means a granulomatous inflammation. So these foci of inflammation that contain granulocyte, and in this case, lots of eosinophils, as well as other innate immune cells, T and B cells, and a sort of focal, somewhat encapsulated inflammation. And polyangiitis means inflammation of multiple vessels. So I mentioned the two ANCA antibodies, PR3 and MPO. In the two other forms, they are present in most patients. In the eosinophilic form, in fact, they are present in less than half of the patients, which most likely is due to the presence of other types of autoantibodies that maybe have more selectivity for eosinophils. Clinically, the diseases are fairly similar. And actually, the logic is really simple. If there is polyangiitis, but no real granulomatous inflammation or foci that you can detect, it is called MPA, the middle form. If there is polyangiitis, but also granulomatous inflammation, you call it GPA. And if in addition to those two things, there is eosinophils present, and typically eosinophilic asthma, then you call it EGPA. So,
Thomas, can I ask you a question? Go back.
Yes, absolutely. Go ahead.
Yeah, go back to the other slide. I don't understand the percentages where it says frequency of ANCA, and you've got P ANCA, C ANCA, and then total ANCA. What's that third category?
Yeah, actually, it's very small print you might see, but the third category is ANCA minus.
Oh, that's a minus, okay.
Yeah. So in other words, in the two first diseases, only 5 to 10% don't have PR3 or MPO or lactoferrin antibodies.
I didn't see the negative sign.
Right.
Right.
So that's the category that then is more than half of everybody in EGPA. And you see what you look for. So there are likely more different autoantibodies that can bind to neutrophils, or in the case of EGPA, to eosinophils, that we just haven't discovered yet. Maybe lower frequencies. And I think, in parentheses, in most autoimmune diseases characterized by autoantibodies, there clearly are probably an order of magnitude more autoantibodies than typically is talked about and recognized. There are some recent larger studies to look for, previously unknown autoantibodies, and there are always plenty of them. Okay, so let's talk about the symptoms of EGPA. So typically these patients have eosinophilic asthma, but otherwise the diseases are relatively but not quite identical. So similar but not identical to the other two forms of ANCA vasculitis. So first of all, there is small blood vessel inflammation. So this is not a disease of large arteries. It's more of small to medium-sized arteries, as well as capillaries, and even a little bit on the venule side of the circulatory system. They can be very few. They can be even a single location somewhere, but they can be widespread and affect every organ in more severe cases of the disease. And particularly with more severe disease, there are the usual symptoms of an active immune response, broad immune response with cytokines being made and so forth. So systemic symptoms like fever, fatigue, weight loss, malaise, just not feeling good at all. Fortunately, for patients who have this form of vasculitis, there is very infrequently involvement of blood vessels in the eye. Otherwise, one of the two big feared symptoms of ANCA vasculitis is blindness, which is simply because small vessels that supply blood to the eye or the optic nerve get inflamed, and there's hypoxia of the relevant organs and tissue damage, in worst case blindness. So very rare in this form, but has happened. Much more common is involvement of the vasculature of the upper and lower airways, which can manifest itself as cough, coughing up blood. And remember, in the context of a chronic eosinophilic asthma, this might not be the easiest thing to recognize initially. May show up on an X-ray initially, and of course, coughing up of blood is not typical of asthma, so that should also raise the red flag that there might be something more going on with blood vessels in the patient. Then there is a range of symptoms. I did say that the involvement of vessels can be in any organ and tissue, and symptoms accordingly can come from any tissue. Arthralgia could also arise just from the systemic disease cytokines. You know, when you have a viral infection, you often have muscle ache and as well as joint ache, and that's partly what could be here. The tissue could also be directly infected by hypoxia and regional inflammation. And the same is true for the mucosal surface, for the skin. Peripheral nerves need a blood supply and can be affected by the vasculitis here. The second big feared symptom of EGPA is a kidney involvement, and typically in the form of a necrotizing crescentic glomerulonephritis, which has a specific histologic picture of it. That, of course, results then in kidney function decline and sometimes hematuria, and eventually, in worst case scenario, results in complete kidney failure and the need for chronic dialysis. I should also point out here that the vasculitis can completely occlude vessels and result in regional hypoxia, which can be very painful. So symptoms can be quite variable, and you can have these regional pain symptoms in various places of the body, including digits and so forth. Briefly about what we understand of the genetic predisposition and what perhaps factors could trigger the disease. So genome-wide association studies in ANCA have done in many diseases, but only, I think, one study of a reasonable size in the eosinophilic form of the disease. And it was quite illuminating in that it turns out that many of the genes, and here's 9 that appear to be important, is somewhat different from the genes predisposing to other forms of vasculitis. And what is really interesting is that a number of these genes, most of them in fact, also predispose to eosinophilia more broadly, and have to do with eosinophil biology. And you can sort of see how eosinophilic asthma could be related to many of these same polymorphisms. So, for example, the BIM protein is clearly important in eosinophil apoptosis, as is a non-coding RNA from a seminal locus. TSLP is a cytokine made by Th2 cells, one that is often talked about as an early cytokine made by epithelial cells to drive a Th2 response. So interesting that it shows up as a risk locus also for this eosinophilic condition. And you can see the others. There's an MHC class 2 association also that's interesting. So clearly what you can see from the genes, that this is a disease that has to do with Th2 immunity, T cell recognition of antigen. And clearly autoantibodies play some role as well. And then eosinophil behavior and egress from blood into tissue appear to be elements of the disease. Interestingly, also, eGPA tends to debut in life a little bit earlier than the other top forms of vasculitis, and as I said, often asthmatics. So the fourth decade is the peak, whereas it is the fifth or sixth decade for the other forms of vasculitis. Anyway, this later in life presentation of autoimmune disease is an interesting aspect to think about. If you are predisposed to autoimmune conditions, why do you get them late in life rather than earlier? And one possible explanation for that is the accumulation of exposures to things such as viral infections, Staph aureus infection, including in nasal upper respiratory regions. Some people think are important in triggering the disease in those that are predisposed. And lots of studies have been done on a number of exposures that may or may not be truly relevant. I think the most likely conclusion is that a number of environmental exposures may accelerate or may predispose it, but no particular agent is clearly to blame. Epidemiologically, for example, patients with eGPA more likely live in rural areas than in urban environments, for example. All of this helps a little bit think about the pathogenesis itself. So the question that I'm mostly interested in is what exactly are the immune reactions and steps that result in this particular disease. So histologically, as I mentioned, the hallmarks are these focal inflammations that typically occur around blood vessels and that contain eosinophils. You may not see it particularly well, but you can see there are these pinkish, reddish cells in pretty good numbers all over the tissue here. And there's quite a few of them over here. So histologically, this disease can be recognized by that if you biopsy the right location. So oftentimes the disease starts in three different phases, which are overlapping in time very much. So usually asthma or potentially other eosinophilic allergic conditions start first. And then over time and in conjunction with those diseases, you have accumulation of eosinophils in tissue, and sometimes you can see them in elevated in blood as well. And then at some point, those tissue eosinophils begin to aggregate in certain locations, and you eventually have a frank necrotizing granuloma forming around the blood vessels. So it is clear, maybe more so in the other forms of vasculitis, that the autoantibodies are instrumental. So they not only are a hallmark of ANCA vasculitis more broadly, but also play a role. So, for example, in animal models, if you just infuse mice with autoantibodies against PR3 or MPO, you tend to get the histologic phenotype of necrotizing granulomas around blood vessels. It's a little bit less clear in the eosinophilic form how this all works.
Can I ask you another question? We're talking about the eosinophil, and yet with ANCA studies, we're looking at autoantibodies against the neutrophil. Do we make autoantibodies against eosinophil peroxidase, against EPO?
You know, that's a really great question. I'm not aware of any literature identifying a specific eosinophil selective ANCA. But, you know, just from the pathogenesis of this form of the disease, I'm pretty convinced that there must be antibodies that selectively activate eosinophils. Now, I've been trying to also look at these pictures as, are there neutrophils there as well? And I think there are some too. So in this disease, it's not like the neutrophils are entirely absent, but, you know, and of course, some neutrophil proteins are also present in eosinophils. They are, you know, siblings, if you will, in the family tree of granulocytes. Anyway, so
Thanks.
yeah, but to your point, most of what we know a little bit better is in the context of neutrophils in ANCA vasculitis, and there's comparatively much less information about the eosinophil. So activation of these cells is clearly part of what goes on, and I gotta say, it's not totally clear, at least to me, what all the factors are that really drive that activation. But the autoantibodies are a plausible component of it, partly because they bind to Fc receptors, which, when cross-linked, can activate all forms of granulocytes. And then this then results in these foci that attract other cells of the immune system, and the activated granulocyte will produce many things, including DNA by etosis or netosis, depending on the type, as well as reactive oxygen species, and the granule proteins, which include enzymes degrading extracellular matrix, and the fairly nebulous eosinophil granule proteins that you know, play roles damaging, or, I mean, they didn't really evolve to damage tissue. They evolved to carry out remodeling and host defense. And as a innocent bystander, you could say normal tissues get damaged. Eosinophils are also good at producing factors affecting the endothelium. So typically, the granulomas foci have endothelial leakage, which promotes the egress of additional immune cells and vascular leakage and local swelling. So these foci often resolve, or at least come to a more dormant phase of repair. What happens depends on the intensity of the initial inflammation. So less severe foci can simply resolve altogether. And typically other mononuclear cells, T and B cells, come in there, monocytes come in there, and you have resolution. But in more severe foci with more dramatic inflammation, more tissue damage, the repair may be delayed or never fully occur. So you may have a lingering long-term chronic presence of monocytes, T cells, B cells, sometimes even ectopic germinal center-like structures, and fibrosis, and what should I say, the unfortunate part of that is that the vessel might be permanently destroyed and occluded, and neoangiogenesis may be able to overcome some of that, but you may have chronic deficiencies in vasculature and hypoxia on a long-term basis, which of course should be avoided to all cost. So the management of the disease, just a few words, not horribly different from the management of many other rheumatological conditions. So the principles are to first try to aggressively treat a newly diagnosed disease, particularly if it appears to be severe and active. And the goal here is to induce remission as rapidly as possible with a high dose of glucocorticoids, typically, you know, a milligram per kilogram. There's a typo, it should be milligram per kilogram, prednisone, alternatively, or sometimes in combination with cyclophosphamide, azathioprine, or more and more frequently nowadays, rituximab. And then as soon as a remission appears to have been successfully induced, the question is of maintaining that remission, tapering down over some period the glucocorticoids, sometimes using IVIG, or increasingly frequently nowadays, some of the other biologics and other therapies that are newly on the market or are still in clinical development out there. And I will talk to you really briefly about the IL-5 class of agents. So the prognosis of EGPA has, as is the case for the other forms of ANCA vasculitis, improved quite dramatically over the last half a century or so. So most patients do quite well and nowadays and remain in remission, or exacerbations can be relatively well taken care of. So really quickly about, this is sort of shifting gears a little bit. I'll tell you what some of the newer thinking in the field of drug development are. So number one, and for full disclosure, 15 years ago, I started what I refer to as a brief sabbatical in the pharmaceutical industry. I spent a number of years developing drugs. I saw this from the inside. And I don't want to influence you in any way whatsoever, just tell you the science of some of the things I saw. So, and in relation to eosinophils, of course. So first of all, the industry is beginning to understand that mouse models, by and large, are not very predictive. So the industry is shifting away from the old idea that you needed to prove efficacy in mouse models before you could enter clinical trials. So that is no longer a requirement, and it never was a requirement by the FDA. It was more for internal conviction. But now it's clear that these models do not accurately, in most cases, reflect disease in humans. They're good for many other things, but not for that. So the industry is more and more turning to studying the real disease in human patients in a number of ways through collaborations or internal work. And as I see it, the real aim of all of that work is to construct a good and plausible hypothesis of what happens in a disease that then can be tested just as a scientific experiment in clinical trials. And so for that, you need to have good ways of measuring the hypothesis and the thing that you are trying to affect with a pharmaceutical intervention. And these measures are usually referred to as biomarkers, and that's an increasingly important topic both for drug development as well as management and diagnosis of diseases broadly. And the biomarkers need to reflect both the key biology as well as the disease itself, ideally, a different set. And the industry is realizing more and more opportunities for a personalized healthcare approach, patient stratification and endotypes and categories that can be predicted. So in the end then, in my mind, a ideal drug candidate ready for clinical trials needs to fulfill these 4 high-level criteria. So number one is, for the industry, very straightforward. The molecule itself needs to be technically great. It needs to do exactly what it should be doing. And this is the one thing that the industry really knows very well how to do. The second thing is a little bit more demanding and, like I said, a newer thinking in the field. Namely, you need to have a clear hypothesis of what that molecule should do. And then number 3, you need to be able to measure exactly that mechanism of action, MOA. And then finally, and more nebulous perhaps, you need to have a good rationale for why that hypothesis fits into the bigger picture of a human disease. So the one drug I saw the development of is called benralizumab. At the time I worked at MedImmune, it was called Medi 563, and I'm only mentioning that because some of the graphs have that abbreviation. So the real experiment is very simple. The question was, do eosinophils that are elevated in diseases like eosinophilic asthma, do they actually play a role in driving the disease? And that's an important question because they might not. But they could even be there to try to heal the pathology going on. And the analogy I sometimes use is that, you know, everything that's elevated is not necessarily bad or promoting disease. So if you think of two aerial photographs from high altitude of a forest fire, one before the fire where the forest is healthy and green, there's no fire trucks anywhere to be seen. But during the forest fire, there are these little red spots, you know, like eosinophils on a immunohistochemistry slide. So these are fire trucks. And they are obviously there to try to put out the fire, even if they are greatly elevated in the condition of the forest fire. So therefore, this question, I think, was important to ask. So the solution to this, the tool for the experiment that was designed in that company was an antibody called benralizumab, which is an antibody specific for the IL-5 receptor, which I told you initially is expressed almost exclusively on eosinophils, and in addition to that on basophils, but nowhere else that we know of. And in order to make this particular antibody more effective, it was engineered to be increased in its binding to the Fc gamma receptor type 3A or CD16, which is expressed on NK cells and many cytotoxic T cells. So in other words, this engineered, otherwise human antibody is very, very effective in binding to killer cells in one end and to the IL-5 receptor in the other end. And so we referred to it as an ADCC enhanced humanized IL-5 receptor alpha monoclonal antibody. So to go back to the drug criteria then, the first criterion to have a technically great molecule. So this was a well-characterized antibody with all the properties and safety profile in non-human primates and half-life and dose and all of these things that would make it ideal for use eventually in human patients. And the hypothesis was super simple in this case. Just let's get rid of the eosinophils. That's what we want to do. And to measure them, it's very simple, already existing assays. You can measure them in blood, you can measure them in tissue. And in the case of eosinophilic asthma, you can take tissue biopsies. You can also get bowel fluid and measure eosinophils in these two locations in addition to blood. And the overall hypothesis was that eosinophils do play a positive role in human eosinophilic asthma. So in this case, super simple idea and a clear experiment to be conducted. And as I'm sure this audience knows very well, a fairly significant portion of asthmatics have elevated eosinophils, maybe something in the range of half or close to. Also, a subset of COPD patients have elevated eosinophils. That's a smaller subset. So the engineering of the antibody, just one slide mention of it. So it turns out that the Fc portion of all IgGs have a carbohydrate in a critical location, very close, part of what the Fc gamma receptors identify. And there's a critical fucosyl residue there. If you remove that residue, which you can, for example, do by manufacturing your antibody in a cell line that lacks one of the carbohydrate adding enzymes, a fucosyl transferase, then the antibody will be made without that fucosyl. And the antibody now binds to CD16 with about 100-fold increased affinity. So here's an in vitro experiment to test this out. So in a mixed culture of eosinophils, basophils with other immune cells, particularly NK cells present, you can see that the antibody in a dose-dependent manner and at super low concentrations, nanomolar concentrations, will result in the killing of all the eosinophils present. And if the antibody is made with the fucosyl, this doesn't really happen very much anymore. So the afucosylation is key to the effective elimination at low dose of this antibody in vitro. And actually, I should say, importantly also, the eosinophils do not get activated and they do not degranulate during this killing process. So this is now the real experiment in human beings. So on the left-hand side, patients were given a single dose of benralizumab in an early study, and at super low doses. So in the first dose, it's 0.3 micrograms per kilogram in a patient. And at all doses, the number of eosinophils in blood dropped down to essentially 0 at the 1 week earliest time point. And then interestingly, if the dose was low, really super low, the eosinophils come back very quickly. And the higher the dose, the longer lasting the eosinophil suppression is. And by histology in a few patients, I think it was 2, the eosinophils also appeared to be entirely gone in the lung tissue. So here's another study also done to look for eosinophils in the airways, and the same idea exactly again, that in most patients given benralizumab, the eosinophils are entirely or almost entirely absent after a relatively brief treatment. So it seems to work. And the real bigger study, the phase 2 study to ask, is that then actually helpful to patients with eosinophils? The answer is yes, it does help them. It's not a cure, but it is a reduction in exacerbations by about 50%. So this resulted in further studies and eventually an approval of this drug. It's now on the market. Here's just another way of showing the same thing. And I should say that there is also another antibody targeting IL-5, actually approved a couple of years before benralizumab. It is a blocking antibody, so it will bind to IL-5, the cytokine itself, and prevent it from binding to the IL-5 receptor. So it's a different mechanism of action, but the end result is still the same. So I told you IL-5 is required for the production of eosinophils. So without IL-5, they aren't produced much. So over a certain time period, eosinophil numbers will decline down to very, very low. So with benralizumab, you have an immediate killing. With the other antibody called mepolizumab, you have a starvation and a slow decline, but the same end result. So let me just summarize everything finally here for you. So I believe that the eosinophil is a very interesting cell with a multitude of biological responses. It's a more complex and versatile cell than I think was initially realized. And it both shares biological responses and patterns with other granulocytes, but it also has a number of unique features and purposes. And it does participate, at least in many diseases, with eosinophilia. And I've told you that eosinophilic granulomatosis with polyangiitis is similar, but not identical to other forms of ANCA-vasculitis. And in this disease, eosinophils really clearly play a role in pathogenesis. And I'll mention in a second why. And I mentioned to you that IL-5 is necessary for eosinophils, and that if you block it or you deplete through the receptor of eosinophils, you will have a beneficial effect in eosinophilic asthma. Importantly, not really any improvement at all in other forms of asthma. And these agents are being trialed in additional diseases. There is a clinical study published in the New England Journal of Medicine using mepolizumab in eosinophilic granulomatosis with polyangiitis, EGPA, and it did improve the disease. So about the same story as in eosinophilic asthma, where exacerbations were reduced by about half. So not a cure, but a clear positive impact and benefit for patients. And I anticipate and looking forward to other eosinophilic diseases being tested to see in which ones eliminating eosinophils will have a therapeutic benefit, or maybe in some of them it won't. Very interesting. So just to show you really quick 2 slides. One of them is that basophils are certifiably eliminated by benralizumab. So here, the 2 lines that end up at the bottom are the 2 doses of benralizumab. Placebo is up at top. And as I mentioned, this would not happen by blocking IL-5, only by killing cells with IL-5 receptors. And the reason is here. So basophils do not need IL-5 for the development, but they do have the IL-5 receptor, explaining what otherwise might have been a mystery. And maybe at some other point we'll talk more about the basophil, which I'm sure you guys know plenty about. So that's it. Thank you for your attention. And I was asked to display the word of the day for those who need to know that word. So thank you very much.
Thank you very much. I don't know who picked the word of the day, but those listening remember it, because after these talks, everybody asks me what the word of the day is, and I quickly forget it. Thank you. That was a brilliant talk. That's I never understood mechanistically why benralizumab was so effective in actually killing eosinophils compared to its competitors, so to speak. We must have eosinophils for a positive reason. Well, we know we have them for that. Do we run no risk of completely wiping out these cells?
Yeah, that's a really great question, and I think that in Western societies, in the developed world, we apparently do pretty well without eosinophils. But I would be concerned to eliminate eosinophils in a person who lives somewhere where parasites and worms and things like that are more common. I have not heard of that type of safety concerns, but I would kind of expect it. And, you know, an interesting parallel to that is that there is evidence to believe that infections by worms and parasites may be protective of diseases like asthma. So our Th2 immunity, and maybe including our eosinophils, do need a little bit of exercise to keep us healthy. And if we don't have it, you know, we develop diseases that are more of a Th2 nature.
Well, thank you. We're out of time. Otherwise, I would solicit some questions, but you can be assured we're going to invite you back because that was a superb presentation for our group. Thanks again.